What's up, everybody? Welcome to Omnipotent Health and Fitness episode number 2. Our guest today is a research scientist at the Institute for Human and Machine Cognition. He is also an associate professor at the University of South Florida College of Medicine, Molecular Pharmacology and Physiology. He obtained his PhD in physiology and neuroscience and his bachelor's degree in nutritional science. However, if you're tempted to just box him in as an academic with no physical prowess, think again. He spends his off days paddleboarding past alligators on his Florida farm and once deadlifted 500 pounds for a set of 10 reps after fasting for 7 days. And he once spent over a week underwater as part of NASA's NEMO mission. So please help me in welcoming the person who many credit for bringing the ketogenic diet into mainstream awareness and who Tom Bilyeu calls the king of keto, Dr. Dominic D'Agostino. Dom, welcome to the show.
Great to be here. Thank you. Thank you for having me.
Thank you. Thank you. So I like to start with everybody's kind of origin story. You know, when you were a kid, did you always see yourself becoming this kind of really famous research scientist working with NASA? How did you get into this?
No, not at all. You know, I wasn't really the best student, I have to say. Although I was very motivated, you know, much more motivated in academics when I started working out. First, kind of like just because my brother was doing it for football, and then, you know, I played football and actually broke my leg, and it became a big part of my recovery for that. Wow. I had a high school biology teacher that really pushed me, and I was in an honors bio class, and that was sort of like the pivot point of, you know, getting more into academia. And I was like, well, I had an interest in biology and nutrition, so I majored in both at Rutgers University. And then my undergrad project, you know, eventually became a PhD project. I studied the neural control of breathing, or respiratory neurobiology, and mostly a focus on neuroscience, but how it's linked with physiology. And then that PhD sort of got me down the path of, I was dive researching at the time. I was really into scuba diving. So I had some grant funding through the Office of Navy Research, which is through the Department of Defense. And they were basically funding me to develop mitigation strategies for Navy SEAL warfighters that are in an undersea environment. And that led me to the ketogenic diet, because seizures are part of that potential side effect. And that was like 2007 or 8. And ever since then, I've been digging into research on metabolic therapies, everything from fasting to ketogenic diets to exogenous ketones.
Yeah, that's amazing. Diving into your work has just been absolutely incredible, because you're touching on all these different aspects of things, but you started with essentially oxygen toxicity leading to epilepsy or seizures underwater for Navy SEAL divers. Can you talk a little bit more about that and kind of the mechanism behind which that would happen and how ketones might help alleviate that?
Sure. Yeah. Thanks for asking. I've had a role at the American Epilepsy Society Special Interest Group in Dietary Therapies, which is the American Epilepsy Society is a very big conference, and it's mostly really funded by the sponsors were the pharmaceutical industry, so anti-epileptic drugs and things like that. So I had a keen interest in nutrition, and I was studying anticonvulsant drugs for oxygen toxicity seizures, which are tonic-clonic or grand mal seizures, the same thing. But drugs really didn't work well, and you don't want to load up a warfighter with drugs because it can dull your performance, cognitive and physical. And I studied nutrition in undergrad, but I think we glanced over the ketogenic diet. But I come to find out that the ketogenic diet is used when drugs fail. And I was inspired by the story of Jim Abrams. Jim being a Hollywood producer, he made the movie Airplane and Naked Gun with Charlie Sheen. And he had a friend, Meryl Streep, who did a movie about the ketogenic diet, First Do No Harm. And I watched that movie in 2007, I think. And then I jumped on PubMed, and I was like, wow, there's actually research. And at the time, it was Dr. Eric Kossoff and a number of other people at Johns Hopkins had, recently published a few clinical trials on the ketogenic diet and validated it as an anti-seizure approach. So my thinking was, hey, I could come back to nutrition. I can get away from drug research, which was producing some interesting effects in our animal models, but not really that great. And I really like this idea of using nutrition and producing a state of therapeutic ketosis to increase performance resilience in extreme environments and to make like the super warfighter, if you will. And as we developed various technologies and diets and even looked at fasting, we saw that we could put an animal in an environment and sustain him in that environment for 5 to 6 times longer than he would otherwise be able to survive that environment. And at the end of that, hour-long exposure, the animal would be perfectly fine in 5 atmospheres of oxygen. So, we're breathing 0.2 atmospheres of oxygen now. Right. So it's like almost 20%. At 1 atmosphere. So the atmosphere that I was studying would be 5 atmospheres of pressure, but at 100% oxygen concentration, which would be 5 atmospheres of oxygen, which increases the partial pressure of oxygen in the brain from 20 millimeters of mercury up to like almost 2000 millimeters of mercury. So it's a very big oxidative hit to the brain. And what we found is that ketones can actually prevent the oxidative stress in the brain. But more importantly was that ketones were able to preserve the membrane potential of the neurons and actually preserve neuronal function in the context of that very high oxidative stress. And it was studying this very esoteric topic that made me realize that this could be leveraged and utilized for other neurological diseases that are pathophysiologically linked to metabolic dysregulation and also glutamate excitotoxicity, which was part of the epilepsy, and pretty much all neurological diseases are pathophysiologically linked to like excitotoxicity, excess glutamate, excess oxidative stress and things like that. And the ketones had worked in our hands better than any other drug, and cocktails of antioxidants and things like that. So it was those observations in 2007 and 8 that actually launched the trajectory of my project for neuroprotection, but then for cancer, and then for, glucose lowering, and then, different rare diseases that we're studying now.
Right. That's incredible. That is just, it's just amazing how the breadth at which this kind of metabolic, when you view things from a metabolic process, how many things that it can touch within the body system as a whole. Before we dive deeper, I do want to stay kind of in around the brain and talk about some disorders there. But before we get too far into the weeds, for anybody that's just not familiar with, they're hearing ketones for the first time, or ketogenic diet for the first time, or exogenous ketones, could you just briefly walk people through the metabolic changes that occur when shifting from using glucose as your main form of fuel to ketones, just what's happening in the body on a pretty big, broad, high level?
Yeah, because I understand that there needs to be more education around this, because I teach at the medical college, medical students, PhD students, and also the undergrad honors college. And there's really not, I think students are taught like nutritional biochemistry, but not really nutritional physiology. And as you probably know, physiology is that's like the foundation of medicine, right? So, and maybe it would be easiest to start with the idea of fasting ketosis. So when we stop eating, 24 hours later to like 48 hours later, we've basically liberated and burnt all the glycogen in our liver. So there's limited glucose availability after that. So the hormone insulin is suppressed, and then we start liberating fatty acids for fuel. And through beta oxidation of fatty acids, we're creating energy, and the central nervous system can't really use the long chain fatty acids that are liberated for energy. So what the physiology does is actually the beta oxidation of fatty acids are converted to ketones through a process called ketogenesis. And beta hydroxybutyrate and acetoacetate are small water-soluble fat molecules, if you will, that enter circulation, and they can cross into the blood-brain barrier through the monocarboxylic acid transporters, the MCT transporters, at whatever concentration they are in the blood. So they freely cross over. And then we get into a state of ketosis, in this state, fasting ketosis. And then the ketones can, after a period of about 4 to 5 days, then about 50% of brain energy is actually coming from ketone bodies, beta hydroxybutyrate and acetoacetate. And then this is, for reasons we don't completely understand, this has a remarkable effect at reducing seizures and probably in some ways improving the efficiency of brain metabolism in the context of ATP generation in regards to oxygen. So the late Dr. Richard Veach did a lot of work in the cardiovascular system and in different cell types, and basically observed that the delta G of ATP hydrolysis was quite higher for ketone bodies relative to glucose. So in that way, you may say that ketones are a superfuel. So reading his papers when I got into this, and the earliest ones were in 1995, I believe, I became very interested in leveraging the metabolic effects of ketones through, well, fasting and then the ketogenic diet. And then it became a real passion of mine to develop exogenous agents that could be, esters or ketone salts or ketogenic fats like MCTs that could be consumed that would produce hyperketonemia. And then this could be sort of a means to augment brain metabolism and overall systemic energy metabolism.
Yeah.
Because at the time, the Department of Defense was not really keen on a high-fat diet for warfighters. So they said, well, try to develop something that, that can be used as a supplement or a drug. So that kind of got me down that path.
Right, using ketone esters as kind of the way to get them into ketosis very quickly. I do want to echo your sentiment. I really wish that we had more nutritional training in medical school. This is part of the reason why it's so exciting reaching out to you, and exciting that you actually agreed to come on here. So we're eternally grateful for that, to just get to learn from you.
Yeah.
Amazing.
Yeah, we don't, I was on the curriculum development committee for the medical college, and as you know, the USMLE or the boards don't really have, you're not tested in nutrition. So the curriculum is so compressed as is. It's not like you can fit in any extra stuff, but we do have a scholarly concentrations in nutrition that I teach, as part of a course for the medical students. And there's different, there's scholarly concentration and in different things. And one is nutrition. I think we have about 30 or 40 students in that I mentor on various projects. And, the students are hungry, no pun intended, for this information. They realize nutrition is important. They realize that if they understand it, that they can make a big impact, later on in their practice.
Yeah, absolutely. Bringing it back to the CNS system, I think I remember back in medical school where we were thinking that epilepsy sort of originated from the hippocampus, which we know more so, more famously as a site of memory. With that kind of loose connection, is there any use to the ketogenic diet in something like Alzheimer's disease, where kind of memory starts to fade?
Yeah. The hippocampus is an area, of memory formation. You got the CA1 region, the CA3, the dentate gyrus. It's a very metabolically active area. So if there's any kind of metabolic stress, that area tends to be impacted because of the high energy demands. It's also the site of very robust long-term potentiation, which is like the strengthening of synaptic activity, and then protein synthesis. So there's like short-term LTP and then long-term LTP that involves protein synthesis and to store and form new memories. So in epilepsy, what happens is that when you have a seizure, and the etiology of epilepsy is largely unknown for many different seizure types, including temporal lobe epilepsy. When a seizure happens, it sort of sends a very robust electrical signal through the hippocampus, and then seizures beget seizures. So when you have a seizure, you're more likely to have another seizure. And the interesting thing about the ketogenic diet, as opposed to drugs, is that when you go on the ketogenic diet, you can wean yourself off, and then you never get seizures again. So that was the case of Charlie Abrams. He would, not everybody, about 10 to 15% are super responders, which means they do the ketogenic diet, the seizures stop for a period of time, and then they're weaned off of it, and they never get seizures again. And that doesn't really happen with drugs.
Why do you think that is? Because of the LTP and maybe some sort of myelination going on, or what do you think?
Well, I don't know. It's a good question. And that actually really got me super interested in the ketogenic diet, that it was almost like in some people a cure for epilepsy. I think what it does is, when we change our metabolic physiology, right, that changes the brain neuropharmacology. And what I mean by that, not only is it changing like brain energy metabolism, and even there's an increase in blood flow to the brain. So the ketogenic diet elevates something called adenosine, and adenosin's a vasodilator. So you can get, increased blood flow to the brain, better metabolic activity, but it also improves, or it decreases glutamate through the conversion of glutamate to GABA through glutamic acid decarboxylase, which is an enzyme that we study in the lab, actually. Interestingly, the ketogenic diet, we know, can increase GAD 65 and 67. Mm-hmm. And when we administer ketogenic agents to laboratory animals, we see an increase in GAD 65 and 67. And the ratio of glutamate to GABA goes down. You have more GABA relative to glutamate. So I think I would say one mechanism could be that you're just lowering an excitotoxic neurotransmitter, which could be glutamate. Glutamate's super important for learning and memory, but too much of it can have the opposite effect. So I think it's balancing neurotransmitter systems in a way that's helping the brain restore network function. And metabolic function and metabolic homeostasis is linked to overall network function, how the neurons communicate in different regions to other regions. And I think by restoring that and maintaining that for a period of time, that can actually prevent subsequent seizures from happening. So in that way, it's like, quote unquote, curing epilepsy over time. So, and then, there's some investigators, all they study is like reactive oxygen species or epigenetic effects and or calcium activated potassium channels, and they say, the ketogenic diet is working through this mechanism, that mechanism, but it's a pleiotropic, which means it's working through many different mechanisms in synergy. And the more I study the ketogenic diet, the more I realize that I'm not going to study just one thing, but I try to study multiple things in synergy, and how elevating adenosine, adenosine receptor signaling is influencing blood flow, which is influencing, other factors.
Things like wound healing too, right? You guys did a study about wound healing, and I'm sure increasing blood flow
right
through adenosine probably works on that pathway as well.
Absolutely. Yeah, you bring that up. Yeah, we got to get that manuscript out. We, one of my students, PhD student, she's moved on now, but she has a fantastic dissertation. And we had one of her committee members, a colleague of mine, was the president of the Wound Healing Society, and developed a very unique aged rat model of ischemic. We would create a wound on one side, on two sides of the animal. One was ischemic and one was non-ischemic, and it was a very elaborate model. But and she had spent her whole life testing things on this model. And then when she came to USF, we put her on the committee, and she's like, well, test ketones. So the ketones, actually, we do a Doppler blood flow measurement on the ischemic tissue, and showed like a remarkable increase in blood flow when the animal was in a state of therapeutic ketosis. Wow. And then what was very interesting is that it worked really well in the aged model, but we also had a young model like juvenile rats, and it actually improved wound healing in the juvenile rats, which was kind of unexpected. And most wound healing strategies are all about topical, like putting things on the wound to enhance that. But when we change our metabolic physiology, that has a much bigger, and people in wound healing don't really think about that. That if you obviously if you lower glucose, then the viscosity of the blood goes down. So you have better perfusion and you're delivering more glucose, less inflammatory factors that are impeding blood flow, and you're elevating ketones, which have a unique role in and of themselves for
Yeah.
wound healing.
Yeah. I think it's amazing. It really does touch on many aspects of the body. And when I was going through framing this interview and my questions, I was thinking I would just go kind of top-down approach, but jumping from one topic to the next is so easy to do because it seems like the ketogenic diet and just metabolism as a whole seems to be able to touch on all of these aspects. Just sticking with the CNS just a touch longer, I know that your wife, and you've talked about it on many podcasts where she's had these studies about how potentially the ketogenic diet could be lowering anxiety. You guys had an observation in your mouse models. Could you tell me more about that? I was really curious about it, and I was trying to dive in and search more. But yeah, could you tell me more about that?
Yeah, sure. Thanks for asking.
Yeah.
Yeah, the first study we did, let's see, it was about, I'm looking at the date, it was over 10 years ago. In 2010, we started gavaging animals with a ketone ester, and then we put them in this hyperbaric chamber where we have sensors on the animal. We get 14 channels of data, and we got a video and everything. And it was observed, a gavage is basically like tube feeding the animal, and then we administer the ketogenic agent, and in 30 minutes, the ketones are at like 5 millimolar, which is like us fasting for a week, right? And then we'd get some baseline activity, and then we pressurize it. And then typically the animal was seized real quick, like 2 to 5 minutes, sometimes 10 minutes. But in a state of ketosis, they would go for like an hour. The first rat was like, wow, we've never seen this before. And then every rat, it was observed doing it. So we published that study, and then we started like formulating different ketogenic compounds and administering them. And my wife was running the studies, which often happens. The PI is so busy writing grants and teaching and doing committee work that they're not in the lab as much. But so she was gavaging the animals and saying, well, the animals are so much more calm when they're in a state of ketosis, and I can handle them. They don't freak out. They don't try to scratch you. They like to be petted. They have an attenuated fear response. And I think we just had like a little bit of money in a departmental account, and they were like, okay, let's study this a little bit with an elevated plus maze and other rodent sort of experiments that you can do. Now, an elevated plus maze is like the rat can go out on a platform that's elevated and be out in the open to the exposed environment, or it can go back into the closed arm of the elevated plus maze. And essentially what we were observing once we gavaged them is that they spent considerably more time, at least when we combined beta hydroxybutyrate salts with MCT. That combination worked better than the ester and a couple other things that we tried. It was a pretty robust effect. Like, it was similar to giving a fairly large dose of Xanax or a benzodiazepine, that it attenuated that anxiety and social anxiety associated with that fear response. So it was very robust. We reproduced it. Not only did we reproduce it several times, then we went and looked at the mechanism, and it seemed to be, in some part, adenosine was playing a role, but also GABA and adenosine, and probably a few other things. But we linked it to those two things. So yeah, she was doing experiments and saying, hey, we need to study this because this looks very interesting. And if we study it, we'll probably get a significant result that we can publish. And another observation we had was working with the glucose transporter type 1 deficiency syndrome mice, the mouthful, when we would put these mice in a state of ketosis, and we were using it to sort of treat their metabolic disorder, it was difficult to put them under anesthesia. We were using isoflurane for anesthesia. So they were like resistant to the narcotic effect of anesthesia, which as divers, this became interesting to us because nitrogen narcosis is a problem that, you know, deep divers have. So another interesting effect of ketones is that it can make the brain so resilient to things that it can prevent sort of the shutdown, the narcotic shutdown of certain drugs. So I said, well, administer the ketogenic agents and then administer the isoflurane and see how fast it takes them to go down. And it was something like, it was pretty big, like 25, 30% increase in time, 50% in some animals.
Wow.
So this is like, you know, no one has ever observed this. So we did a whole study and we published it in one of the bigger anesthesia journals, the effects of ketones, you know, basically preventing, and that could be serious for someone undergoing anesthesia, you know, at least with isoflurane. So we have yet to actually do all the different anesthetic agents, but it was, you know, all animal researchers use isoflurane. So that's another observation we made.
Right. That's interesting. Anecdotally, as soon as literally when you sent back the email that said, yeah, sure, I'm down to do this interview, I went ketogenic that day and I'd never tried it before. And I also have a 10-month-old at home who like keeps waking up, and prior to the ketogenic diet, and again, it might just be placebo or whatever, but you know, I'd wake up at 2 a.m. I'd be way more chill when I was in ketosis than when I wasn't. And I don't know if that's due to ketogenic diet or being in ketosis or not. And I did the Keto Mojo and I was at like 3.2 for my ketone levels. So
that's pretty high. Yeah, you're definitely in ketosis. Yeah.
Well, I I was consuming too much MCT oil. I was lacing my coffee with it, and I think I hit like 9 tablespoons, which ended up being like, I don't know, it's like 800 grams of fat, and had some GI upset. But I think that was about the max that I could tolerate GI wise.
Yeah, that's pushing. You can build a tolerance to MCT over time. And if you incorporate it and administer the MCT with like food, so you could make like salad dressings, mixing MCT with extra virgin olive oil, and then, you know, consuming it that way. Yeah, I work with different dietary teams that work hard to get ketones elevated in kids with epilepsy. And there's many different strategies, and MCTs are great. They're like the poor man's ketone ester, and they work remarkably well. And it's nice because it's like, it's, you know, it becomes endogenous ketone production through, it's a ketogenic fat, and your liver is making the ketones from high beta oxidation of these fatty acids. So actually the MCTs combine well with ketone esters or ketone salts. So that combination is actually very good to get maximum ketone levels. And you can avert the GI side effects and use less MCT.
Yeah, and I've heard MCT powder tends to be a little bit easier on the GI as well.
For sure. Yep.
Just kind of one more thing that's a little bit more selfish of me, and then we can get more to like probably what the audience wants to hear. But for me, so, you know, I heard you mention Thomas Seyfried, Dr. Seyfried out here, and I reached out to him, and so I'm also going to be interviewing him in a little bit. But
great,
so his idea about, I mean, it's not specifically his idea, but he wrote an entire book about cancer as a metabolic disease, and you know, which led me into looking up Warburg Effect, and you've spoke about it on many podcasts. And I was wondering if you could give, you know, myself and everyone else a quick primer about the Warburg Effect and how that has to do with the ketogenic diet and cancer metabolism.
Yeah, and you know, I did not know of the Warburg Effect prior to studying different cancer cell lines. So to take a little bit of a step back, before looking at the ketogenic diet, the main focus I had was developing hyperbaric technologies. And one was a laser scanning confocal microscope inside a hyperbaric chamber, which allows you to optically section cells and actually look at the mitochondria in real time, you know, under different conditions. And another technology was atomic force microscopy, or AFM. So we took an AFM and designed a hyperbaric chamber that it goes around. And I started looking at brain cells, muscle cells, human dermal fibroblast cells, and U87 MG cells, which were glioblastoma cells from a 44-year-old patient. And what I saw here is that when I hit the cells with high oxygen, they overproduced massive amounts of reactive oxygen species, and the cells were dying faster relative to normal cells. And the mitochondria were looked to be defective in some way. They were generating more superoxide anion, which is the precursor reactive oxygen species, can then form hydroxyl radical, which can oxidize membrane lipids, proteins, nucleic acids, and things like that. So I was studying this and I didn't really understand what I was observing. I didn't know why the mitochondria in these cells were overproducing oxygen-free radicals, and the mitochondria were exploding with high oxygen. The cells were exploding, and I thought this was kind of interesting. And then I started getting interested in ketones and noticed that when I tried to grow these cells in the presence of ketones, they did not grow well. And I thought my lab tech was doing something wrong. So I approached her and I was like, okay, we need to figure out what's going on here. And then we would take the ketones away and the cells would grow fast. We would put ketones in and the cells would grow slow. So one thing led to another. And then Thomas Seyfried published a paper in 2010 in Nutrition and Metabolism called Cancer as a Metabolic Disease. And I read this paper, I read it over and over, I studied it. It was a very new idea to me. And then I called him and he said, what you're observing in these highly aggressive cancer cells is the Warburg effect. So which is defective mitochondrial oxidative phosphorylation and compensatory fermentation, which is an upregulated glycolytic activity. And they grew like crazy, if I put sugar in, and then if I impaired hexokinase, which is a glycolytic enzyme, with 2-deoxyglucose or lonidamine, that killed all the cancer cells. And if I took primary dissociated hippocampal or cortical neurons and put in glucose and then put in ketones, the ketones seem to be very neuroprotective for them. But if I took the glucose away and kept the ketones, the neurons were fine. But the cancer cells, if I kept the ketones in and took the glucose away, all the cancer cells rapidly died. So this was very interesting to me, and I realized that there could be something to study here, and that really nobody was studying it. So Tom Seyfried, long story short, became a collaborator where we did the ketogenic diet with hyperbaric oxygen and ketogenic agents. But the Warburg effect is essentially damaged mitochondrial oxidative phosphorylation. Over time, all the different carcinogenic substances, you have radiation, you have chemotherapy, you have chemo is, a carcinogenic, hypoxia, all the oxidative stress, inflammation, hypoxia, radiation. There's many different things that can cause cancer. And this is called the oncogenic paradox. Mm-hmm. And it was a Hungarian that actually coined that term, that many different agents can cause the same thing and be an oncogenic stimulus that can induce carcinogenesis. So, my understanding is that this was damaging the DNA, and that DNA mutations would then trigger oncogenes, and then you'd, get cancer from mutations. But what the metabolic theory posits, and it's still like a theory, but it's becoming more accepted, is that the mitochondria are selectively vulnerable to carcinogenic agents, even viruses. So I asked Tom, well, what about viruses?
That's what I was going to ask. Directly. That was one of my questions that I was going to ask him. Yeah, go ahead.
Yeah, like the Ralph sarcoma virus, like there's, there's human papillomavirus and Epstein-Barr even, I think. So viruses actually will target mitochondria more now that as our knowledge about viruses have increased, and Tom always was sending me all these papers, and he's like, no, viruses will target preferentially the mitochondria. So the mitochondria have DNA, and the DNA repair mechanisms of the mitochondria are not as robust as the DNA repair mechanisms in the nucleus. And as we are exposed to these carcinogenic agents, whether it be viruses, radiation, different chemicals, say we're drinking alcohol or drinking some kind of substance, and take the liver, for example, we're stressing it in a way that impairs mitochondrial function, causes mitochondrial oxidative stress, and those reactive oxygen species can also cause cancer. But essentially what happens is that it causes an energetic crisis within the cell, which means ATP levels fall. And then there's a retrograde response of the mitochondria to the nucleus. So the nucleus senses that the cell has impaired energy production. So what it does is it kicks on a variety of different genes, some of them being oncogenes. Some cells die, and some genes are activated that can confer protection. And then it's at this intersection. It's a dynamic interplay of, there's many different things going on. I don't want to oversimplify it, but that will initiate the transition of a normal cell to a cancer cell. And it can be highly different depending whether we're in the brain or in the kidney or in, muscle, but muscle is pretty resilient. So essentially what the Warburg effect is with damaged respiration, you get impaired energy production in the cell and an energetic crisis is sensed. And the DNA repair mechanisms in the nucleus are highly, highly sensitive to the energy status of the cell. So if the ATP levels fall, then DNA repair does not happen at the level of the nucleus. It's super energy dependent. So what happens is that the fidelity of the nuclear genome is compromised in the context of an energy crisis. And then this can then exacerbate and fuel DNA mutations that can then trigger mutations that trigger a normal cell to go into a cancer cell.
Gosh, that's so interesting.
That's kind of like it. Yeah. It's super simplified and it's much more complicated than that, but that's the most simple way I know how to explain it.
No, I appreciate that. I mean, it's what we can do in the given limited time that we have.
Yeah.
And, just as a quick, it could be quick, but it could also be a very long answer, whichever you prefer. But I know I heard Dr. Seyfried talk about a pulse press kind of protocol in treating cancers, and you're focusing on GBM, glioblastoma multiforme, but it sounds like, listening to some of his interviews, that it's across the entire cancer spectrum. Are there any studies done on this already? I mean, it sounds incredibly fascinating.
Yeah, and so Dr. Tom Seyfried and I, we agree on many things, but there's some things that we sort of disagree on, which is really good. You don't want to agree on everything, and you actually want a lot of people to challenge what your ideas and your theories, which many people do. I would say pretty unsuccessfly. They have been challenging Tom in many different ways about his theory. So I'm of the understanding that cancer is a metabolic disease, but it's probably also a genetic disease, and there's a dynamic interplay, and it's a metabolic, epigenetic, and genetic, and there's a lot of things going on there, and immune-based things. So the press pulse protocol is essentially creating an environment where you are metabolically putting very high stress on in particular, very aggressive, rapidly dividing cancer cells. So if you limit glucose availability with a ketogenic diet, suppress the hormone insulin drives, mTOR and IGF-1 and other growth factor signaling that is intimately associated with the growth and aggressiveness of many types of cancer cells. So many people think about, starve the cancer of glucose. Well, I think that's just like a small point. Like, limiting glucose availability will definitely limit glycolytic flux, which will limit not only the energy, impair energy production, but also the biosynthesis, because the glucose is used to create other biomolecules. But I think really important, I think of the ketogenic diet as insulin suppression therapy. So it's suppressing the hormone insulin, and it's also elevating ketones. And in our hands, if we keep everything the same, glucose, insulin, everything, and just add ketones, what I was seeing in some of the original experiments was that it was impeding the proliferation of the 3 different cell lines, cancer cell lines that we were looking at. So that sparked my initial interest in it. So the idea then with the press pulse is to implement a ketogenic strategy that lowers glucose and elevates ketones. And Tom likes to use the glucose ketone index. in millimolar concentration, that would be if you could lower your glucose from like 4 or 5 to 3 and elevate ketones to 3, that would be a glucose ketone index of 1. And we know from the world of epilepsy and seizures, independent of the etiology, a glucose ketone index of 1 has a very strong anti-seizure effect. So for someone with a brain tumor, that could be super important to block the seizures associated with that. When you're in that state, then you can apply other things that would be like hyperbaric oxygen therapy, would increase oxidative stress. And because glucose is low, that would decrease the pentose phosphate pathway. So you couldn't make as much glutathione. It would selectively elevate reactive oxygen species in the cancer cells relative to the healthy cells. And this is what we see experimentally. And there's things like intravenous vitamin C that can, be a pro-oxidant when it's delivered IV and drive things like the Fenton reaction. In a tumor, you have cells that are growing, expanding, but also a lot of cells that are dead and necrotic. And that tends to release a lot of heme proteins, which has iron. And in the presence of high concentrations of vitamin C, that can drive something called the Fenton reaction, which could drive hydroxyl radical and actually kill the tumor cell from the inside out by increasing oxidative stress more. Then there's metformin and other modalities too.
And before anybody goes out and doses a bunch of oral vitamin C, it needs to be IV, right? And so, don't just, eat a bunch of vitamins.
Yeah, oral vitamin C is completely something different. When you take vitamin C, you actually rapidly downregulate the transporter, so you can only achieve micromolar concentrations of vitamin C, whereas when you deliver 25, 50, or 100 grams of ascorbic acid, vitamin C, intravenously, you are actually going into the millimolar concentration. So it's many fold higher. And vitamin C is an antioxidant. When you take it orally in the body, it functions as an antioxidant, but at really high concentrations, it can actually function as a pro-oxidant in some ways, and also a glucose antagonist. So vitamin C and glucose share the same transport mechanism. So when you're delivering vitamin C, you could be potentially impeding glucose transport, which could then trigger a big energetic crisis in highly glycolytic cells. So this is, we know this experimentally, and I don't know how it's been validated, but I mean, mammalian physiology is mammalian physiology. So this inspired, I had a great undergrad honor student actually do a project with vitamin C. She went on to Oxford to do a master's, and now she's at the Moffitt Cancer Center doing her PhD in redox biochemistry and some other things. She's not, I want her to pick up the vitamin C stuff, but she's mentored by a different person now, which is good. You want to move on and to different, actually, she's being mentored by Dr. Lou Cantley's protege, one of her, who's at the Moffitt now. So, which he could be another interesting guy. I highly recommend interviewing Dr. Lou Cantley. I would love to. Yeah. So okay. So the press pulse is basically the press would be the state of therapeutic ketosis, glucose ketone index of 1. And then we have other modalities that would be pulse modalities. And this could be hyperbaric oxygen therapy, intravenous vitamin C. I am also in favor of using the standard of care chemotherapy, radiation, and immune-based approaches, which would also synergize. And also metabolic drugs like the PI3 kinase inhibitors and actually they tend to work if you administer a PI3 kinase inhibitor through a counter-regulatory mechanism. It tends to increase insulin, which is a bad thing. But in the context of a ketogenic diet with suppressed insulin, that sort of unleashes the cancer-killing effects of PI3 kinase inhibitors. At least this is, I think, observed in animal models. So it'd be a good question for Dr. Lou Cantley. So, and also I think it's, when we talk about chemotherapy, it can be toxic, but I think of chemotherapy as also as a form of, and radiation too, as a form of immune therapy. When you're killing cells with chemotherapy, you get like cell lysis syndrome and you get autophagy. and the cell lysis syndrome and autophagy, once those little bits of cells and things enter circulation, then that actually stimulates the immune system, and the immune system becomes hypervigilant. So we know that the ketogenic diet can increase cancer-specific immune regulation or vigilance. It can help the immune system find cancer cells, identify and neutralize them. And then when we deliver ketogenic agents, we're killing cancer cells, and you could have something like a toxic tumor lysis syndrome. But I think it needs to be appreciated too that once if you could do a lower dose of chemo, for example, kill some cancer cells, and then the dead cancer cells and tumor lysis also augments this is not really talked about, but I've been talking with different people about this, and they're getting me to think about standard of care in different ways. And I think it can be done in a way that's synergistic with metabolic-based therapies, but also chemo and radiation can produce tumor lysis syndrome and autophagy that can activate the immune system. And of course, there's many different immune agents or agents that can augment the immune system. There was a recent paper on PD-1 inhibitors. So these are like checkpoint inhibitors, right, and then they tend to synergize well with the ketogenic diet. A nice study came out a few months ago on that.
That's so interesting. Yeah, just honestly, I wasn't even looking at any of this stuff until I started diving into your things, and then went down the rabbit hole, and it's so incredibly fascinating.
Well, I'm not a cancer researcher.
Yeah.
So when my graduate student at the time, Dr. Angela Poff, came to me and wanted to do a cancer project, I was like, I don't know anything about cancer biology. So I had to self-learn everything. I know physiology, I know neuroscience, and I know cell biology. So my cell biology background, I needed to get back into that, the whole book, The Cell, and reread that, and then read the biology of cancer, which didn't even talk about the Warburg effect, but right, now they included it in it because that was a major flaw in the book. But then I had to relearn a lot of things. Yeah. And the more I got a big appreciation for leveraging and how we could alter metabolism to target, cancer growth and proliferation. And I was really amazed that like people were not doing this at the time. This is like 2008, 2009. But now there's like whole conferences on cancer metabolism. And now there are whole conferences on, targeting nutrition. It didn't exist. And I think Tom Seyfried reinvigorated sort of the community, who's still very skeptical. They sort of believe that the Warburg effect happens, but they feel that Warburg was wrong. They feel that genetic alterations then lead to the Warburg effect, and the Warburg effect just occurs to direct energy metabolites to biosynthetic processes. So it's like the cancer cells are rewiring metabolism to basically produce this Warburg phenotype of the cells, and then that phenotype helps the cancer cell, grow and survive. And the metabolic theory is basically you damage the mitochondria, and then it's the insult to the mitochondria that creates the oncogenic transformation of the cells. That's the initial cause of the cancer, is that hit to the cellular metabolism.
It's this chicken or the egg phenomenon type thing. I'm reading Tripping Over the Truth right now, and it's a very interesting read, and they're talking about that whole cycle. But to switch topics just a little bit, you talked about autophagy, and oftentimes people combine autophagy with fasting. And so when I was looking up the ketogenic diet and trying to separate out the pros and cons of fasting versus ketogenic diet, they work on very similar pathways. And so I was wondering, what is the difference between fasting versus the ketogenic diet? And is there a situation where one is better than the other?
Yeah, good question. There's a lot of overlap. At the very basic level, you're suppressing the hormone insulin. And then, but in the context of fasting, you're creating energy deficiency, and you're creating an energy deficit and a larger suppression of things like mTOR. You have a bigger activation of AMP kinase. Insulin is lower. Although you could do like a fasting modified diet or fasting mimicking diet, and while you're doing that diet and you measure insulin, it's pretty low and almost similar to fasting if you do it correctly. And Dr. Walter Longo has done some research on this. And a clinical ketogenic diet that's like 90% fat, if you measure insulin, which is something I did quite often just to see, it's on the low end of normal and even below normal sometimes. Like it looks like you're fasting. If you were to draw blood from a person who's on a ketogenic diet, and it's eucaloric, it would look very similar to someone who's fasting. And when you do a well-formulated ketogenic diet and calorie restrict it, and you draw blood, it looks very similar. You would have a hard time convincing a metabolic physiologist that the person's not fasting. You'd have to look at like blood levels of amino acids and things like that. So in many ways, although when you tie it, you asked about autophagy, we don't really have, and we're studying that now, but we don't really have a good tool. We don't have like a commercially available autophagy meter. Right. So you have to look at things like the autophagosome, like LC3, and different ratios of these things over time. And I think it varies depending upon what tissue you're looking at, whether you're taking it from the blood or taking it from the muscle. So autophagy is very interesting to us, and we have a couple projects on autophagy, but it's easier to, and I don't know, autophagy in the brain probably is not very robust as like the muscle and the liver, but we're collecting organs from animals under different protocols, and I think we'll gain some insight into that. My feeling is that when you fast or do a calorie restricted ketogenic diet and achieve a glucose ketone index of 1, you are kind of maximizing autophagy. It's almost like maxed out. Probably not a good state to stay in unless you have epilepsy or cancer or something like that. But I think periodic ketosis is very helpful. Chronic ketosis is probably not so much a natural state to be in all the time, but I do think you're maximizing autophagy in that state.
Right. Kind of shifting gears more towards implementing the ketogenic diet for people who are thinking about doing it. For the, I guess, still on the intermittent fasting versus ketogenic diet, I know with intermittent fasting, some people do like a 5 and 2, they're fasting for 5 days and then like five, like 16 hours in those 5 days and then 2 days off. Is there anything like that for the ketogenic diet, or would that be sort of counterproductive to what you're trying to do if you just do like the weekends, you're just eating normally, and then like 5 days during the week, you're ketogenic?
Well, context is important. And so it depends on why you're doing the ketogenic diet. And if you're doing the ketogenic diet to treat a condition that the ketogenic diet is used for, like different metabolic conditions and epilepsy, over the years, I've communicated with many epilepsy patients, seizure patients, and also neurologists and dietetics teams that tell me their patients now recognize time-restricted feeding and have incorporated it into their ketogenic diet regimen. And it has been the difference of, they do a ketogenic diet and then they get occasional breakthrough seizures. It doesn't work for everybody, but it works for about 2/3 of the patients. But if you take those patients and do intermittent fasting with them, then they can reestablish seizure control. This is very common. So I think there's good therapeutic synergy between when adding time-restricted feeding to a ketogenic diet. And I think this could be important for, Alzheimer's disease, for, cognitive function, perhaps even in healthy subjects, maximizing autophagy, maximizing your fat burning. We've done a little bit of research where, especially in aged animals, if you do low carb or ketogenic and then you do intermittent fasting, that triggers like this metabolic switch. So you get higher fat oxidation, in the context of that. And I do believe there's also a metabolic memory, like when you work out, you build up to a certain strength level, take time out, get back into it, you quickly get back up to that strength level again. And when you train your metabolism to oxidize fat and make ketones, and then you transition off of that and you get back on it again, you start producing ketones real fast, which means you're oxidizing fat fast, because ketones are byproducts of fat oxidation.
Right.
So we see this like in animal models, and likely happens in humans too. So I think people need to experiment depending upon if they're using the ketogenic diet for glycemic control. Like I wear a continuous glucose monitor, and my glucose trace looks very, very boring. It looks like a flat line. So that tends to translate into not only high fat oxidation, but just stable energy level and flow throughout the day, which from my perspective, it makes me productive in the context of working in academia. I just have like a more steady, mental flow, I guess you could say. So on the weekend, sometimes I do kind of take off. I almost do this 5-2 thing that you mentioned, right, where I'm strict during the week. And then on the weekends, if we're eating out or something like that, I'm less strict, but pretty much always low carb, but not like clinical ketogenic.
Right. Awesome. In regards to, I kind of want to talk about some of the difficulties people have in even just trying the ketogenic diet. So one of the things that I hear about, like my wife's a nurse practitioner, she says people try ketogenic diet, they come in and then their LDLs skyrocket like 6 months later. And so she was like, if you're going to do this, make sure you're checking your LDL, like you need to go set up an appointment. What's your experience with that? Or, is there any research being done in that regards?
Yeah, what we see, so we're doing a clinical trial now with subjects, and we do see that elevation of LDL. What I can say is this, is that if you have a patient coming in that's really like insulin resistant, overweight, and borderline hyperglycemic, like type 2 diabetic, and you put them on a ketogenic diet or a low carb diet, a lot of times their LDL will improve. So a lot of times, or it'll stay the same or it'll slightly increase. On the other hand, and this seems to be pretty consistent, if you take a normal, healthy, athletic individual and they're on a high carb diet and you put them on a very low carb, high fat diet, I have seen LDLs triple and even quadruple. And it seems to be this lean mass hyper responder phenotype, or whatever you want to call it. So that term is being used. And you see other biomarkers of metabolic health improve, like triglycerides go down, HSCRP stays the same or goes down, LDL or HDL will often go up. But the LDL, sometimes LP little a, ApoB, these things are correlated, not always, but usually will go in the direction that's not atherogenically favorable, I guess you could say.
Right.
And that happened to me, I would say, to some extent. And one way that I reversed it was to go from eating a lot of dairy fat and a lot of fatty red meat to more eggs and a lot of fish. Right. Eating a lot of fish, a lot of olive oil, a lot of avocados, nuts a little bit, but nuts too many don't agree with me. So basically reducing the saturated fat and getting proportionally much more monounsaturated fat, but also adding some carbs back in the form of fiber. So eating, broccoli, asparagus, cauliflower, a lot of arugula, salads, green leafy vegetables. I have a salad pretty much every night. So I think the added fiber and reducing saturated fat to like 20% and getting a lot more monounsaturated and a lot of omega-3 fatty acids really helped get those numbers pretty much back to normal. I have borderline high LDL, but it actually cut in half just by doing that. Yeah. But originally, I was following a very restrictive ketogenic diet, very high fat, and even restricted in fiber and vegetables to achieve that fat level. But now I'm probably about 60% fat, maybe 30 or 40% maybe about 30% protein, with the balance being fibers, carbohydrates.
Right. I think that's where I'm at too. Obviously I've only been doing this a few weeks, but I pretty much don't restrict any green leafy vegetables I want. I eat all the asparagus and broccoli I want. And then, just supplement with, meats and fats, the MCT oils. I didn't realize how hard it was going to be to find healthy fats that were easily consumable, I guess. I just assumed that it was everywhere, but it really, you got to look for it. But,
yeah. And then, meat is kind of, I don't eat as nearly as much beef as I did, but almost every day I have some. And I mean, we live on a farm, we have cows. There's like cows all around us, and all of it's all grass-fed, purely pasture-raised beef. So I have access to that and buying it wholesale just from all the cattle ranchers that I know. But even though I have access to that, I tend to gravitate towards a lot of fish. I eat quite a bit of fish and eggs. And I give a lot of the yolks to my dog. So I'll have 8 eggs this morning, but I had 4 yolks, and I gave 2 yolks to each of my dogs, and then mixed up their food. They basically eat what I eat. And then I, will have quite a bit of olive oil, MCT oil throughout the day. And I think I'm having ribeyes tonight, so steak. So
I think we're having ribeyes tonight too. So that sounds
great.
The last subject that I'm actually mortified that we're sort of crunching on time, because there's so much I want to ask you. But I think the one thing that I'd be remiss if I didn't talk about was a ketogenic diet in regards to weight loss. And I've read some studies that were saying, they compared ketogenic diet. And what I'm unsure of is whether their ketogenic diet that they did was like the traditional ketogenic diet or like this modified Atkins type of thing. But I guess their big thing is that the rebound effect of like gaining weight after, because they say that the ketogenic diet is unsustainable, they're unable to do it. And so it seems to me on the surface level that the ketogenic diet would be excellent for weight loss. I mean, I was trying to maintain my weight, and I went from 170 to 160 in a week, and I was eating a lot. And I'm really active and I work out, but I dropped 10 pounds really quick. So I was just wondering what your thoughts were in regards to the ketogenic diet and weight loss.
Yeah, so I do believe in the calories in, calories out, with the caveat that there's other things going on, I'll say. Of course, I really believe that, higher protein and higher fat have a satiating effect on the brain, and they change brain chemistry and in ways in most people. Not everybody that follows a ketogenic diet is going to lose weight. The caloric density of a ketogenic diet is quite high, like 50% higher calories. So if you put the same amount of food on your plate and you're not eating as much vegetables and everything like that, you could easily get a caloric surplus. So if you're on a ketogenic diet and you get off of a ketogenic diet and you gain weight, I would say you're gaining weight because you are not in a calorie deficit anymore. You're in a calorie surplus. So you're likely what happens when you transfer from a low carb or ketogenic diet to a higher carb diet, then it has a hedonic effect on eating, and eating carbohydrates can elevate dopamine in ways and increase the appetite control centers of the brain, where you just eat more because it's more hyperpalatable. A ketogenic diet is, I would say, sort of hypopalatable. It's hyper-satiating and hypopalatable relative to the standard American diet. So right. You inadvertently eat less calories, whether it seems like it or not. Like in the long run, most people lose weight, and simply due to the calorie deficit that it produces, because ketogenic foods are not as hyperpalatable, I believe. Although right. Yeah. Eggs are like super tasty to me. Like a can of sardines is almost like eating candy for me. If I'm hungry, it's very satisfying and satiating, and I love the foods, but there's only so much that you can eat, right? Whereas if you're eating fries or eating potato chips or pretzels or something like that, it's easy to eat, overconsume.
Right. Absolutely. I mean, when I say I was eating a lot, it's because I was eating platefuls of broccoli, and that like really fills you up, and getting like multiple plates of broccoli. But even then, that's going to be less than, a Big Mac or something like that. So, yeah, but yeah, totally.
Well, I do think that, so I had this question quite a bit with the carnivore. I get so many questions about the carnivore diet and everything. I do feel that it's not a great idea to just, go completely meat only, the paleo keto diet or whatever you want to call it. There's just so much good data on fiber, on epidemiological data. You could criticize it, but the data is just so overwhelmingly strong that fiber correlates to all these better health outcomes. So why don't we get the best of both worlds? We do, high fat, moderate protein, and high fiber. I think it can be do. So that's sort of the diet. I eat much more fiber now than I did when I was on a high carb diet and just eating. I grew up Italian, just eating bread and pasta and things like that. And would always have an egg white omelet. Maybe I would throw a yolk in here and there, but always threw my yolks away. I can't believe I did that. But but yeah, I think we can get the best of both worlds by maybe not necessarily having to go completely ketogenic, but low carb, moderate protein. But there are many benefits to being in a state of ketosis. By definition, elevating your urine, breath, or blood ketone levels. We know that ketones are powerful signaling molecules that have epigenetic signaling effects on the brain and things like that. It's a big part of the research that we do. So I'm inclined to, if you're not on a ketogenic diet and it's low carb but not ketogenic, I think there's a lot of benefit to including things like MCT oil, ketone salts, ketone esters. I put in the realm of like medical conditions or military conditions, but ketone salts taste good. They're available. MCTs are great. I think they're versatile. You can include them in cooking and things like that.
Yeah, absolutely. Again, we're running out of time, and I really wish that we could talk more about all these things. I wanted to touch more on like the NLRP3 inflammasome and epigenetic
Part
pathways.
2.
Yeah, if you're up for it, I would be more than thrilled to have you back on. But before I ask my last question, where can everybody find you and follow your research and your content?
Yeah, thanks for asking. Ketonutrition.org. So ketonutrition.org. We have a blog, we have a newsletter. Try to put a lot of information on there. I have some former PhD students that write, some med students that write for it, and then we co-write articles together on topics that are interesting and relevant, and I think that can inform people. There's a lot of misinformation out there, so I'm trying to be a website that can cut through the BS and give people information that's actionable and they can apply to themselves.
Yeah, absolutely. And I can attest to that. It's a wealth of information and beautifully arranged. And I used it quite a bit in preparation for this interview.
Great,
So,
thanks.
Yeah, of course. So this last question, I like to keep it a little bit fun, a little bit more quirky. So it's the Magic Genie question, but you only have 2 wishes. One of your wishes is for the entire world to adopt one lifestyle that would most greatly impact their health. And the other wish is extremely selfish, and you can't save the world, can't cure anything, strictly for yourself. What would those 2 wishes be?
Oh, strictly for myself, 2
So
wishes.
one of them is for yourself, one is for other people to adopt one, lifestyle modification that would most impact their health.
Lifestyle modification. Yeah, you know what? I think, and I'm not gonna like go right to diet, right? But I think when you like define your purpose, like you are interested in medicine, you can make a difference and impact people's lives. I would say spend time to reflect every day on your purpose in life. And the more you understand your purpose, then the more you're going to be internally motivated to sort of make a difference. And the more you can sort of introspectively sort of meditate and think about that purpose, then you can you'll be internally motivated. You won't have to use, I think, a lot of drug use, a lot of behaviors, and a lot of problems are created in society just because people do not pursue their purpose. And so that would be, for people need to carve out, I know you're super busy, but carve out creative downtime where you literally put in your calendar and say, I'm going to go for a walk. I'm not going to listen to my music or anything. I'm just going to think and think about my purpose. And I think that's super important. And it's become increasingly more important, as I advance in my career to do that.
Absolutely.
To carve out time to do that.
Yeah, I love that.
Oh, and selfish. So selfish things that I would do. So the question, reframe it like,
what would you wish for yourself?
For myself? Yeah. Kind of hard. I feel like I have everything. I'm very grateful. I'm doing what I love to do. But time, right? So I wish, academia has pros and cons, but you have to, like in any job, you have to allocate a significant amount of time doing things that you would otherwise not want to do, like BS, obnoxious administrative stuff. So I kind of wish I had more time to play. For me, playing is just like being out on the farm this morning. My parents are visiting, so we got out on the ATVs. I have three-wheelers, like these vintage three-wheelers. So we were running around with the cows. I couldn't find the cows, but I found them over in the pasture. So, and that was just like, that just sets me on fire for the rest of the day. It's just so fun to be outside, be in nature, be around animals. And that playtime is, I don't always get it. Yesterday I worked like a 14-hour day, but today I loved it and was like, man, I need more of this.
Yeah, absolutely. That sounds incredible. All right, everybody. If you guys haven't dove into his world, it's completely amazing. I think you're known as the king of keto, not necessarily just for your amazing research, but I mean, I went head on into all of your content and I literally couldn't complete all the different podcasts that you've been on and everything like that. And I can tell that it's very important to you to get this, information out there. And I'm eternally grateful for you taking the time to be here with us today. So thank you so much, Dom.
Thank you for having me. I appreciate it. And love to do part 2 if you're ready.
Yeah, of course. I'll reach out. I'll give you some time to have that downtime and to play. But thanks so much.